All right, so the first one we want to go over here is structure number 1, the purple stuff with the brown dots. What's the purple stuff with the brown dots? Rough ER, right. All right, and what does the rough ER do? Okay, transports protein. Well, it doesn't have to say where, just transports protein. Okay, will be good enough. All right, so 1 mark each for that. 1 mark for rough ER, 1 mark for transports protein. All right, structure number 5, what would that be? That is the nuclear membrane. Okay. And what does the nuclear membrane do? Julia? Out of the nucleus, right. Yeah, controls what gets in and out of the nucleus. That's good enough as a function for that. If they have something like controls what gets in and out of the nucleus, that would be fine. Okay. All right. Number 6 is pointing at the little dots. Some of them are on the rough ER and some of them float free in the cytoplasm. What are they? Ribosomes. And what do ribosomes do, Brooklyn? Right, they make protein. Right, so if they have something synthesize protein, make protein, something like that, give them a mark. All right, number 7. James, what's number 7? The Golgi apparatus. And what does the Golgi apparatus do, Jace? Right, gets rid of wastes. Okay, excretes wastes, something like that. Something to do with getting rid of waste. Yes, that will work. Okay, number 9. Keandra. Mitochondria, yep. Okay, that's an R, not an H. It's just really bad. Chondria. That doesn't even look any better. Okay, what does the mitochondria do, MJ? Okay, it has to do with energy. What are we? Burns what specifically? Sugar. Okay, burns sugar to make energy, right? They do have to have something in there about breaking down sugar, burning sugar, something like that, okay, because energy is very general, all right, because there are actually 2 forms of energy that are dealt with by the mitochondria, the kind it uses and the kind it makes, all right? So we can't just say energy. We have to say uses this form to make something else. No, because there are other products of the lysosome. Okay, the lysosomes also make amino acids and things like that from breaking down your food. All right, number 10, lysosomes. All right, and what do lysosomes do, John? Right, they digest food. They're the stomach of the cell. Okay, they're the things that have enzymes in them to break down your food into simpler materials like amino acids, sugars, okay, things like that. All right, number 12. What do we call stringy things that would run throughout the cell, Colton? Well, microfilaments, I'll also accept microfibers. I would even, let's say today, accept microtubules. All right, so any of those, all right, any of those micro things, microfilaments, okay, microfibers, microtubules. Okay, they all do the same thing, which is that they, Maddie? The stringy things. Right. Supports the cell, keeps it from rupturing, keeps it from stretching too far. If they have anything in there essentially about stretching, keeping shape, supporting, I'll accept that as the answer. All right. And then lastly, okay, we have these 2 things, which are what, Dacia? Centrioles. Okay, and as Maddie was telling us just a minute ago, the centrioles are responsible for making sure that DNA gets to the right side of the cell during cell division. So if there's something like helps with cell division, secures DNA or something like that, okay, give them a mark for the function there. And then give them a mark out of 16, I think, right? We had 8 of those. So give them a mark out of 16. Let them see it. Put it in the box. No, no. The ribosomes make the protein. All right, so we'll go over structure of DNA, history of DNA, and then we'll also look at the differences between prokaryotic cells and eukaryotic cells. We go into a bit more detail in AP on that material as well. Okay, so first thing, we need to understand how the structure of DNA makes it capable of storing genetic information. Okay, now, in DNA, you always have these things called base pairs, right? A always goes with T, and C always goes with G. You guys remember that from Science 9? Okay. Now, those are the only possible combinations. A with T and C with G. Only 2. What else uses only 2 things to code information? No, I'm thinking something nothing to do with living things. Binary code. Your computer uses series of zeros and 1. Okay, 0, 1, 0, 1, 1, 0, 0, 0, 1, 1, 0. That's all. That's what your computer views data as. Okay, binary code. It's on or off, 0 or 1. Okay, can we put a lot of information into a computer using only zeros and ones? Yeah, which is why DNA works just fine too. All right, it's got 2 base pairs, and as long as you change up the order of those base pairs along the strand, you can make that sequence code for lots of different things in the same way that a computer sequence of zeros and ones does exactly the same thing. All right, you guys follow me there? That's how DNA works. All right, is it simply uses the sequence of AC, or sorry, AT, CG, you know, all the way down to code for things. All right, secondly, learn how the structure of DNA was discovered. Kind of an interesting story about 2 heavy drinkers who decided to pair up and discover something important. Okay, no, I'm actually serious. Okay, evaluate technologies related to DNA based on their uses and their implications. So we'll look at a few of the sort of practical applications, okay, of DNA stuff. All right, so evidence that DNA was the genetic material of cells. The biggest thing that got this started was people watching cells divide. Okay? The process of cell division goes through the same steps every single time a cell divides, no matter what kind of cell it is. All right? The nucleus begins to darken, okay, as cell division approaches. The reason it begins to darken is because the DNA is being condensed into chromosomes, and it's being copied. So there's going to be twice as much of it, which is kind of what makes the nucleus get darker. There's going to be twice as much DNA in there as there is normally. Okay? Then the nuclear membrane begins to disappear. And these chromosomes begin to line up along the middle of the cell, called the metaphase plate. Right? And then the centrioles go to the ends, if it's an animal cell. If it's a plant cell, obviously that doesn't happen. And 2 copies of all of the DNA get to each side. All right? But the biggest thing there was in every single cell they watched, the first thing that happened was the copying of the DNA. Right? Would that seem to imply that it's important? Yeah, because there was no copying of mitochondria, there was no copying or doubling of rough ER, all right, or anything like that. None of the organelles get essentially doubled before cell division. Why do you suppose the cell doesn't bother doing that? John? Exactly. Okay, the new cells, when they're done dividing and they split in half, can use their own DNA to build a new mitochondria or a new Golgi apparatus or anything like that. They don't need to necessarily do that before the cell divides. They'll be able to do it because they have the instructions to do so. Right? So that led people to think that whatever was in the nucleus had to be pretty important. Okay? They watched what happened with the stuff that was in the nucleus, even though they didn't really know what it was. Okay, but they got the idea that it had to be pretty important. Okay, all right. So you can sort of see here in this diagram that that's what's going on. This is a cell that's in what we call interphase, which is the normal life cycle of the cell. It's what cells look like most of the time. All right. During prophase is when the DNA is getting copied and is starting to condense into the chromosomes. Okay? In late prophase, you see the centrioles going to the two ends, okay? And you see the chromosomes beginning to migrate towards, okay, the metaphase plate, because during metaphase is when they're all lined up. Okay? Everybody follow me there? All right, so DNA is made up of countless pairs of bases. The order and arrangement of these bases is what codes for materials and characteristics that the organism has or requires. All right, so the bases are adenine, which we abbreviate as A, thymine, T, okay, cytosine, and guanine. All right, now you can see in this diagram here, okay, we'll say that, we don't know for sure, but we'll say that blue represents A, okay, red represents T, orange represents cytosine, and green represents guanine. All right, so you can see that they may not always be, it might not always be cytosine on the left and guanine on the right, but it's always cytosine and guanine together. It's always adenine and thymine together, right? And there's really, essentially, an infinite number of combinations for those things to have, okay, along the DNA molecule. Now, does that mean that every single person's DNA is completely different? No. All right? There's a lot of stuff that all of our DNA has that is exactly the same. All right? Especially things that are just metabolic processes. All right? I mean, the way that your cell makes mitochondria is essentially identical to the way my cell would do it. All right? That part of the DNA for both of us is going to be pretty much the same. All right? It's other things that are heritable characters that would be a lot different. All right? And that's why when they're doing the CSI kind of DNA test, they're looking for specific markers. They only usually look for about, I think it's about 35 or 40 different markers, okay? And those are ones that are almost always different between individuals, right? But I mean, obviously, a lot of the metabolic stuff is the same. And in fact, if you were to look at most mammals, a lot of their DNA, okay, would have similar sequences to ours because, again, a lot of their metabolic processes are the same as ours. Okay. All right. So early on in the study of DNA, it was discovered that, okay, through chemical testing, that the amount of adenine was always equal to the amount of thymine, and that's what led people to the idea that these two bases must pair together, okay, and the amount of cytosine, always equaled the amount of guanine. So they figured if they're always present in the same amount, they must go together, all right, which was a logical assumption. Okay. Now that helped scientists to deduce the shape of DNA, because these bases, adenine, thymine, and whatever, they have different shapes, and their molecules have different sizes. So in order for this idea of the double helix, okay, to be true, A had to go with T and C had to go with G, otherwise there'd be bulges in the helix all the time. All right? If you untwist the DNA helix, it looks like a perfect ladder. All right? It's the same distance across all the way down. Everybody follow me on that? Okay, but if you paired A with G instead, you'd have a ladder with a bulge, okay, and then a ladder with a bulge, and it wouldn't look right. It wouldn't be this neat sort of, delicate shape. It would just be kind of random, and that's not what we're looking for. Okay, since we know that A and T always have the same amount, C and G always have the same amount, it made sense that they must go together, and that kept the shape. All right. Other evidence that DNA is species specific, okay, in the DNA of any one organism, the amounts of the four bases are not all equal, and the ratios vary from one species to another. All right, so what we're looking at here are what we call karyotypes. Okay? Karyotypes are when you essentially take photographs of different of cells during cell division while the chromosomes are lined up at the metaphase plate. All right, so the one on the right is fruit fly DNA. These are the chromosomes that fruit flies possess. Okay? They don't have, obviously, as many as we do. They're quite a bit simpler. All right? So they don't have nearly as much DNA. All right? And so you can see there are markers along here, okay, telling them, that marker number 1, who knows what it codes for, could be eye color, okay? Marker number 2 could be wing shape, because there's all kinds of stuff. When you do genetics in university, you will work with Drosophila all the time, okay? Drosophila melanogaster is the technical name for fruit flies, and they are your best friend while you are working in genetics, because you can breed them easily, you can and they reproduce quickly. All right, you can within a week have a whole new crop of fruit flies to gas and then check for different characteristics. All right, so you spend a lot of time over the dissecting microscope. Okay, brick eyes, brown eyes, red eyes, sepia eyes, curly wings, normal wings, striped body, bland body, and you have to sort them all. It's a pain in the neck, but okay, it's one of the things you do when you're investigating Mendel's ratios and stuff that we talked about the other day. All right, so fruit fly DNA has all been marked. They know what part of every chromosome codes for whatever characteristic they're looking for. That's why you see all these little markers in there. All right, for a human karyotype, okay, you'll do this as well. Okay, in probably your second year of genetics, they'll bring in a community health nurse. She will take your blood. Okay, and then the next week you will look at your blood sample under the microscope, the white blood cells in particular. Okay, and you will look at them dividing, and you'll take pictures of each and every chromosome. All right, in your cells, and then you'll build your own karyotype. Okay, that's what this is. That's me. Okay, how can you tell it's me? You can't, except you can tell it's a man because it's got an X and a Y. All right. So when you're looking at this, okay, you can see there's 2 copies of each and every chromosome, and each and every chromosome does have a very specific shape. Some what we call telomeres in the middle. Okay, some of it's closer to the end. Some of them are longer than others. Okay, things like that. Those are the things you have to look for when you're deciding. Oh yeah, that's chromosome 4, as opposed to chromosome 20. Okay, you can see that the larger numbered chromosomes are actually quite a bit smaller. All right, so you can tell the differences based on that kind of stuff. You'll learn that when you get there. Okay. So does that make sense? Okay, 2 copies of every chromosome. Here's, I don't know which one was maternal or paternal. There's no way to know that. Okay, but you could say this is like the maternal one, and this is the paternal one, maternal, paternal, okay, and so on. But you always got 2 of every single one. Okay. All right, questions there? It's actually kind of cool when you do this and you see your own. Okay. All right, so most biologists agree that DNA was the genetic material, okay, but there was sort of a race to prove that this was true. All right, no one had actually ever gotten a picture of DNA because obviously it's a molecule. I mean, even though it's a giant molecule, it's still small. All right, and there was, certainly chemical testing that proved certainly that A always goes with T and C always goes with G, but somebody had to find the shape. There were lots of theories on the shape. Most were that it was a strand. Okay, that was more like a ladder, right? But Watson and Crick, okay, are the 2 guys that actually got a picture of DNA, okay, proved this double helix shape that people had been theorizing. Now, Watson and Crick were 2 alcoholic scientists. They did a lot of drinking. They met in a bar, okay, and they decided, after they'd had probably too many, that they should work together, which was odd because no one wanted to work with either one of them. Okay. Probably because they drank and weren't very nice. I don't know. Okay, but anyway, this partnership, although celebrated, was very brief. Okay, these guys didn't get along when they weren't drinking. All right, and so obviously you can't get a lot done when you're drunk all the time. So this was a very short partnership. Okay, but anyway, what they did is they came up with some ideas, and then they performed what's called X-ray crystallography. So they did a chemical process we won't go into in detail that causes DNA to crystallize. And then they took an X-ray photograph of it. And when the DNA crystallized, it held, okay, it kind of made it get stiff so that it would hold the helix shape as opposed to wrap itself around itself, where then it would be difficult to surmise its shape. It actually caused it to straighten, okay, and stay in that rigid form. All right, so that was their idea, was that if they could do this, they knew that crystallography was a way to get a picture of big molecules, so they tried it with DNA, okay, and this is what they got. That's Watson and Crick's picture of DNA. All right, it's an X-ray, a microscopic one, okay, of the top of a DNA molecule, looking down on the helix. All right, so you can see that it X's through, all right, and that there's this ring around the outside that's faded here on the edges, but dark here. All right, and so that led to the idea that if you were looking down a spiral staircase, this is what you would see. Agreed? Anyone ever look down a spiral staircase from the top? Right? The railing kind of gets you know, because of that perspective effect, it gets kind of closer together as you look at the bottom, in the same way that railroad tracks look like they go together in the distance. Okay, well, the same idea looking down on a DNA molecule from the top. All right, you got this, the effect of the spinning made some parts appear dimmer than others, and it gave you this idea that they crossed in the middle. They don't really cross, but because you're looking down on a whole bunch of them twisting through each other, it looks that way. Okay, so this proved, okay, kind of unequivocally the shape of DNA for Watson and Crick, and then after that they decided they didn't like each other anymore. Okay, and that was the end of the Watson-Crick era. Well, era, a month and a half, whatever it was that they worked together. Okay, but still pretty important in that they got something major accomplished. All right, so Watson and Crick based their model of DNA on the data they were able to extract from those images. Okay, from those images, they also knew the scale. Okay, they knew that it was approximately 1 nanometer. Okay, from the center of the helix to the edge. Okay, and that the distance between a certain number of pairs was 3.4 nanometers. Okay, and the distance between one pair, okay, was 0.34 nanometers. So they were able to kind of figure all this stuff out from their pictures. All right, and that said to them, well, that means that A definitely does have to pair with T, and C has to pair with G. Otherwise, we won't have this nice distance across being consistent. All right, you guys follow me there? All right, it was when they saw that picture, they're like, there's no bulges in this. It's almost perfectly uniformly circular, around the outside of the helix. That means that there can't be any bulging of the molecule anywhere. All right, and so that's what led them to the idea of this helical shape, this double helical shape. Okay. All right, so essentially that's what we just said here. It's just another picture of it. Okay. No, the A will just simply not bond with G. It won't bond with C. It just won't happen. Okay. Mutations aren't bulges in the DNA. Mutations are reordering of the sequence of the DNA. All right, so no, a mutation isn't going to cause base pairs to pair up the wrong way. It's going to cause them to pair up in a different order. All right, and in the same way that if you have a computer program that says 0101 and it's a series of steps, do this, then do this, then do this, if your DNA gets out of order, just like a computer program, the computer program runs differently. Right, and then whatever your DNA was supposed to build in that section that's been mutated is built differently, better or worse. Okay. All right. Okay, so the bases were either too wide or too narrow to be paired with themselves, okay, because there was this idea that maybe adenine just bonded with itself and cytosine bonded with itself, okay, because they knew chemically that that was possible, all right? But again, that would have led to parts of the molecule being wider than others, and that didn't fit with the uniform shape that they got, okay? So that's when this was sort of confirmed, this A with T and C with G, and then that gave rise to the, well, if you've always got these pairs, a sequence of pairs could code for something if the sequence is different in every person. All right, so the ladder-like structure of DNA and the fixed pairing of bases led Watson and Crick to that conclusion we just mentioned, okay? There was no rule to the sequence of the base pairs along the DNA strand, meaning the strand was like a combination lock with a nearly infinite number of possible sequences. Okay, so A, T, C, A, right? This just, it almost looks random unless you know what you're looking for. Okay, and that's what the Human Genome Project, which ended now probably about 10 years ago, okay, was trying to do. The Human Genome Project was mapping the human genome. And that means essentially looking at all of the DNA present in a human being and figuring out what every section of it did. Okay, pretty big job? Yeah, because there's a lot of stuff that your DNA codes for in a complex organism like ourselves. Right? But it's done. Okay, we now know what every part of every chromosome of every strand of DNA in your body does. A lot of it is filler. A lot of it is stuff that's just holdover, okay, from, you know, our ancestors that we don't, it's just useless DNA. It's kind of miscode. It doesn't really do anything. All right, and that's part of the research that's going on now is, does that have any relation to certain types of diseases that humans can develop that aren't necessarily caused by anything, they just develop in people? Could that be part of this holdover, okay, DNA that we still have that doesn't really appear to do anything? Okay, and so that's where a lot of the research is now. Okay, so that was the key mechanism for heredity, okay, and that's when people started looking at, you know, this southern blot idea where, okay, here's a parent and here's their child. Look at how many markers they have in common. It's about half. Okay, and you do that with the mother and the father and then the two children or whatever, and you could see, oh, half of each. Well, that definitely confirms this idea that genetic material, that DNA is genetic material. All right. Okay. All right. Okay, now, copying of DNA. Once this helical ideal, the double helix, was confirmed, the mechanism for copying DNA just kind of fell into place. People discovered it really, really quickly. It's very easy to copy DNA. All right. All you do when you're about ready to have a cell divide is you just rip the DNA into its two strands. Okay? Your cell literally pulls the DNA apart and then just starts putting the complementary pair on the side of each strand that's left. Okay? And so you got one strand going to one cell, one strand going to the other cell, and all that your cell does is simply build the other side again. Right? Because it's easy, because A always goes with T and C always goes with G. You only really need one side. All right? But this is the mechanism for making the copying go a lot smoother. All right? So what your cell will do, okay, is it will pull across or pull apart, okay, with this enzyme called helicase. Okay, you're not going to have to know that. Okay, helicase pulls the helix straight. Okay, so it removes the twist. Okay, pulls it straight and then separates it. And then you've got DNA polymerase. Okay, DNA polymerase starts to add the other strand. Okay, and so that gives you this double-stranded, and then by the time this all gets to the end, okay, you'll now have these two strands, two copies of everything, and the cell can then effectively divide. All right, so there's several different kinds of DNA polymerase. There's kind of the one that does the building, and then there's the guy that checks the work. All right, so DNA polymerase 1 is sort of the spell checker. Okay, it goes along behind and makes sure there's no mistakes. Okay, yeah, and well, a more accurate spell checker, yeah. Okay. All right. So what Watson and Crick actually wrote in their paper was, we've confirmed that the shape of the DNA molecule is a double helix, and we don't want you to copy our idea, so we're going to tell you this. It has not escaped our notice that the specific pairing we have postulated immediately suggests a copying mechanism for the genetic material. They didn't know what it was, but they didn't want anyone else to get credit for it. That's the way these guys kind of thought, right? It's like, look, we figured this out. Don't take credit for it later. Right? I mean, someone else figured out exactly the mechanics of it, but they were the ones that said it could be done, just someone else will figure it out. Okay. All right, so Watson and Crick had discovered meant that only half of a DNA molecule, okay, or essentially an RNA molecule, was needed to decode the information. That's what led to the idea that protein synthesis can come from the DNA molecule, or from simply one side of it. So while your cell, we talked about this before when we were talking about the parts of the cell, while your cell is in interphase, it's normal functioning, okay, if it needs to build something, it just pulls that section of the DNA apart, copies one side, and sends the copied one side molecule out to the ribosomes, because again, you only need to know one side of the molecule to build whatever it is you need. It's complementary. Okay, making sense? All right. All right, now, that means that sometimes the ribosomes will get one side of the DNA molecule. Sometimes they'll get the copy of the other side. Does it matter to them? No, it's always this sequence, right? It's the AT, CG, 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 AT, AT, right? It's always going to be, if I got an A, I know I had 3 C's in a row. Or if I got, 3 A's in a row, I had 3 T's in a row, okay, and so on down the way. It doesn't matter what side they got, they still know the sequence. Okay. Also, obviously, that double strand means it's easy to unzip, is the word they use. Okay, the molecule, and get at one side. All right, and so this is sort of the process of this replication. Okay, you got DNA ligase, which kind of puts everything back together. Okay, you got the pulling apart. Okay, the replication fork is what they call right here, where it's being pulled apart. Okay, things like that. All right, so that's all you need to know about DNA. Again, I'm not going to be asking you about names of enzymes like ligase, polymerase, okay, transcriptase, or any of that. Okay, just so you understand, it's a double helix. A always goes with T. Basically, same stuff you learned in Science 9. Okay, and I'd probably ask you about Watson and Crick because I think they're funny. Okay. All right, so we'll go over here, prokaryotes and eukaryotes, okay, differences, importance, effects, okay, that being a prokaryote has over being a eukaryote. All right, what kind of cells do we have again? Eukaryotic cells, right? That might have been important for your cell anatomy lab for one of the variables. Okay, we have to be able to identify and describe the characteristics of prokaryotic cells, okay, versus eukaryotic cells, and then obviously distinguish. Distinguishing between the two types is very easy. One has organelles and the other doesn't. All right, so distinguishing between them is pretty easy. You guys not have this one in there? Did I forget to put it in the notes package? I probably did. Okay. Don't worry about it. We're just going to go over it. Okay, and there's not going to be any of it on there. We just want to go over, summarize here. All right. Okay, so there's three shapes, three classes that prokaryotic organisms, okay, can appear as. All right, there are the spheres, okay, and the spheres are called cocci, okay, C-O-C-C-I. Right, so if you've ever had strep throat, how many people have had strep throat before? That is caused by Streptococcus pneumoniae, all right, a sphere-shaped bacteria that can infect your throat. The flesh-eating bacteria, also a coccus bacteria. It's a mutated form of strep throat, actually. Okay. There are the rod-shaped ones called the bacilli. So if you've ever eaten at Arby's and gotten salmonella, that's what you had. Sorry, I'm not a big Arby's fan. I've only eaten at Arby's twice, and both times I got salmonella. So, not a real big fan of Arby's. That's why I always bash them when I teach this lesson. So you got bacillus bacteria. So yeah, salmonella is an example of that. Okay, tetanus. Okay, also an example of a bacillus bacteria. You've had vaccinations for that throughout your whole life. If you step on a rusty nail, you go get a booster. All right. They can obviously be quite toxic to you. Right? Salmonella mostly infects you and affects your digestive tract, right? Whereas tetanus will affect your muscles. Right? Anyone know what happens in tetanus? Okay. What's the other name for tetanus? Kind of a common name they used to call it way back in the day. Heard of lockjaw before? Okay. If you ever got tetanus, people would literally break their own jaw, all right, from having tetanus. Because what tetanus does, it releases a toxin that interferes with the way your muscles work, and it doesn't allow them to relax. So if you clench a muscle, it won't relax. You clench it again, it clenches harder and won't relax. So people would eventually get to the point where they would clench their jaw so hard they could break it. Okay, like crush their teeth. Yeah. All right. That's how they would, essentially near the end, you would die because you couldn't breathe anymore, because the muscles that control your breathing would be constricted and you wouldn't be able to ventilate your lungs anymore. All right. Yeah. It wasn't a very good way to go. All right. That's why you get vaccinated for it, so that you won't die that way. Yeah. Okay. And the last one are the spirals, called spirillae. I think that's two L's, spirillae. Okay, and they're the spiral-shaped ones. They're less common. You don't see a whole lot of them. Syphilis is a spiral bacteria. Okay, it's a sexually transmitted disease that makes you go insane. You laugh. I'm serious. I would never tell you something that wasn't true. Okay, there's a lot of historians that believe that people like Napoleon, Josephine, whatever, they all eventually succumbed from the effects of syphilis, causing their neural processes to not be good anymore. That's why a lot of people think Napoleon lost the Battle of Waterloo, okay, because he wasn't thinking straight anymore. It's gotten so bad that he just went crazy. All right. What's that? There is that, but I thought it was something else, but I've heard that several times too. Yeah. Okay. Like I said, it's a sexually transmitted disease, right? And so a lot of sort of royalty way back when, who may not have been so good about their behavior, okay, may have contracted these types of diseases. Okay. All right. Other thing about bacteria is they all have cell walls, but these cell walls are not like plant cell walls. Okay. They're analogous, which means they're a wall, okay, but they're not homologous, which means they're not the same. Okay, Plant cell walls are made out of cellulose. Bacterial cell walls are made out of different materials. The most common one is something called peptidoglycan, okay, which is a long kind of chained sugar, right, that these things are made out of. Penicillin breaks down and reacts with peptidoglycan. So when you take penicillin, it will destroy the cell walls of the bacteria that are infecting you if their cell walls are made of peptidoglycan. And then your immune system has a much easier time killing the bacteria. All right, so that's what penicillin will do. It essentially strips the protection from all of the bacteria and they die fairly quickly. Well, when you ingest penicillin, the molecule that the mold secretes is toxic. I mean, we call it penicillin. That's not the actual name of the molecule. It's the name penicillin comes from the fungus that produces it, penicillium. Okay, and so this penicillium fungus produces a chemical that essentially keeps bacteria from growing on its food source. Right. So when you see something that's got penicillium mold on it, there will be almost no bacteria on that food source because the penicillin's killing it. Right? So we ingest that material, our body doesn't break it down, it gets into our bloodstream where the bacteria are, and it just kills the bacteria that way. All right? That's kind of how all antibiotics work. Right? Problem is that if you have any one of these that is slightly different, then what does penicillin do to it? Nothing. All right? So you kill off a whole bunch of bacteria that are what's the word I'm looking for here? Wow. No. Anyway, a lot of bacteria that could be killed by penicillin. Okay, they die. You leave one left because it's immune. What does it do? It reproduces, and you get a reemergence of essentially the same infection or a different infection because this stuff didn't get killed by the penicillin. And so what that's produced over a few generations of penicillin use is a bunch of penicillin-resistant bacteria. All right, which is why now we have to take stronger antibiotics a lot of the time, like streptomycin, okay, and things like that, that are a little bit harder on your body, okay, but are needed in order to kill penicillin-resistant bacteria. All right. Okay. The other big thing here is that they don't have any defined nucleus, and they essentially have no membrane enclosed organelles. Okay, few, but almost none. All right, so the shapes, like we said, okay, the cocci, the spirilla, and the bacteria, and the bacilli. All right, so tetanus causes lockjaw. The coil-shaped ones, okay. Treponema palladium is the bacteria that causes syphilis. Okay, Streptococcus mutans, okay, can cause strep throat, okay, and things like that. All right. Now, bacteria, just like eukaryotic organisms, are sensitive to similar things to us. Okay, they're sensitive to salt levels. They're sensitive to temperature. All right. But it doesn't mean that all of the prokaryotes are sensitive to the same levels as we are. Right? We have found bacteria growing in the most inhospitable places on Earth. Okay? We have found them in lakes that are under kilometers of ice in Antarctica, okay? Surviving in horrifically cold conditions, and they're thriving. Right? We have found them living in hot springs, almost the exact opposite, where the water is 80 degrees Celsius, okay? And they're thriving. It's not only really hot, but it's also really saline, full of salt, okay? And they survive very well in these conditions. They survive in places that you couldn't conceive of a eukaryotic organism surviving. Some of them are just adapted that way, and because they're so simple, are less sensitive to those changes. No, we have not confirmed the existence of bacteria on Mars, no. There's a lot of theorizing as to where it could possibly be located, but there's been no confirmed discovery. No. They found water, yes. Okay. They found that the water contains certain types of chemicals conducive to the growth of bacteria or life forms of other kinds, but they've actually found none of the actual evidence of the bacteria themselves. All right. Now, obviously, the bacteria that grow in a hot spring are probably not of a real concern to us because they wouldn't survive in our bodies because the conditions aren't right for them. Okay? If a bacterium that could survive in 80-degree water is put into a 37-degree Celsius body, it's probably not going to do very well. It's adapted to grow in different conditions. So those aren't ones we really need to worry about. We need to worry about the bacteria that are at home. okay, in conditions like what we need. All right? Are there ways to kill those? Yes. They're the same things that would probably kill us. All right? So that's why we do things like dry and salt meat if we want to keep it for long periods of time. Right? If you've ever made jerky, right? The reason you make jerky is you dry it out so there's very little water, and then you fill it up with salt. Because if you make it really, really salty, okay, the process is called curing, okay, then bacteria can't grow in it. Okay, smoking does the same thing. All right, smoking the meat, not smoking cigarettes. Okay, if you put the meat in a smokehouse, you get the temperature up, you dry it out, it fills up with salts and kind of oils from the woods that you're using to smoke it with, and those will also prevent the growth of bacteria within the meat for at least a few weeks. Okay, and then it doesn't have to be refrigerated. All right, other things that we might do, we put in preservatives. Okay, now the big controversy obviously on preservatives is anything that would kill bacteria is toxic. We probably shouldn't eat it either. All right, I mean there's a certain amount of logic to that, but there's also a, well, how much of it do you actually need to eat in order for it to really harm you? Okay, so there's kind of good and bads to preservatives. Okay, obviously we can use bleach. Okay, if we need to kill off bacteria on, let's say, a cutting board. Okay, by the way, that's what this picture is. That's a scanning electron micrograph of an uncleaned cutting board. Okay, this is fungus, bacteria, fungus. All right, you definitely want to clean your cutting board between uses. All right, and not just with soap and water, because when you cut on it, you make cuts in it. All right, and stuff gets in those cuts because as you cut, you force the material with the knife into those cuts. All right, you need to soak cutting boards in between uses, especially if you've cut anything like chicken or meat or any kind of stuff like that. You definitely need to bleach it. All right, a cutting board you cut vegetables and fruit and stuff on, not as big of a worry. All right, but still should clean them. Soak them in bleach every few uses. But every time you cut chicken or meat on a cutting board, you should bleach it. In fact, should you use the same cutting board for both? No. Okay, not a good idea unless really you're bleaching it every single time, and you just never get it all. All right, so something to be cautious of. Now, you've heard me several times talk about petri dishes and growing bacteria on a petri dish. That's what this is. Okay, this thing here is a plastic petri dish, and what's in it is something called agar. All right, agar is this magical concoction that you can make that is essentially an all-you-can-eat buffet for bacteria. Right? It has all the things they need to make protein, so it's got nitrogen and oxygen and stuff like that. It's got sugars, lots of glucose in it, and then you just add a little bit of gelatin to it to make it solidify. So it looks like Jell-O, but it doesn't taste like Jell-O. It doesn't taste very good, in fact. It's got this really strange taste because agar, the stuff that has all the nitrogen and stuff like that that they need for proteins in it, has a really funny taste. All right, but what you do is you swab something, okay, and then you swab it onto the petri dish, and then that's where it grows, and that's where you can see here. You can actually see where the swabbing was done, kind of back and forth. All right, you can see the bacterial colonies that are growing from that. Every one of those little circles represents where you had one bacterial cell on the petri dish. Right? If you've ever had to get a throat swab, anyone had a throat swab before? Okay, that's what they did. Okay, they swabbed your throat and then they took it into the back in the lab and swabbed it on the petri dish and stuck it in the incubator. All right, and 24 to 48 hours later, they pulled it out and went, all right, we got some colonies. They take a little sample, smear it onto a microscope slide and look at it under the microscope, and they can determine by adding a certain kind of dye whether we can make you feel better with penicillin or whether we can make you feel better using something else. All right, it's a pretty simple process. That's why it doesn't take all that long. Most of the time it takes for a throat swab to come back is waiting for the bacteria themselves to grow. All right, okay, other things we want to do, obviously, to keep ourselves healthy, boil unclean water, okay, because the bacteria, again, that would hurt us are sensitive to temperature. And if we heat something up really good, we'll kill them off. All right, cooking meat thoroughly, okay, obviously, if Arby's doesn't cook their meat enough, okay, and you get salmonella because it hasn't been heated up enough to kill it off. All right, that's why you probably don't want to eat a blue rare steak. Okay, anyone ever seen a blue rare steak? They literally walk it through a warm room. okay, and set it down in front of you. Blue rare means they do this. They throw it on the grill and they go tss, tss, and put it on your plate. That's blue rare. They can say they put it on the grill, and then they're not legally liable for your sickness. Okay? Ever seen anyone eat one? It's a little disconcerting. I like rare. Don't get me wrong. It should when I cut into it, but okay. If you've ever seen somebody eat blue rare, it's actually kind of disgusting because there's a lot of blood and liquid on the plate when they're done. Like, eat your baked potato first, or it'll be swimming. Okay. Yeah. All right. Now, like we said, bacterial walls are analogous to the plant cell walls. Okay, they serve protective and supportive function, but they're made of stuff called peptidoglycan. So there's two kinds of bacteria. There's gram-positive and there's gram-negative. All right, gram-positive bacteria have peptidoglycan cell walls. All right, they're the ones that can be killed by penicillin. So if you looked at them, you would see their cell membrane. That's this part here. And then you would see this peptidoglycan, okay, on top of it. And that's a pretty protective thing. It would protect it from your stomach acid and enzymes and things like that. All right. But like we said, penicillin will break it down. Now, when you put this dye on that I was telling you about, if you ever had a throat swab, okay, the bacteria will appear purple if they have peptidoglycan in their cell walls, because it reacts with the peptidoglycan and turns purple. All right. If you have bacteria that are gram-negative, the gram stain doesn't absorb, and they turn red, or stay red, okay, as the case may be. And that's because they've got a membrane, a small amount of peptidoglycan, and then another membrane outside of that protecting them. Okay, so penicillin will do absolutely nothing to those types of bacteria because it cannot get at the peptidoglycan through the membrane. Okay. Making sense? Questions so far? Now, lots of bacteria also are soil microorganisms, and they're really important in terms of cycling nutrients, okay, returning material back into the various cycles, okay, especially the nitrogen cycle. All right. Nitrogen is probably the biggest or most key ingredient in fertilizer. Okay, so in fertilizer you have things like nitrates. Okay, you have some ammonium, this stuff here, right? Because that's the stuff that your grass can turn into protein, because protein has lots of nitrogen in it. All right, and so that's why we want to put that stuff on our grass. But we've got bacteria that will break certain forms of it down and turn it into other forms that can be absorbed by plants. Okay. All right, we're going to skip over ammonification here. All right, and so there's also some insects, okay? These are called sow bugs, right, that live in like compost and things like that, and they have bacteria that actually live in their digestive tract in the same way that we do, okay? What bacteria lives in your digestive tract normally? Okay. It's okay for them to be in your large intestine, but if you ingest them and they get into your stomach and small intestine, you get really sick. E. coli, all right? This is why if you ever see someone who's a cook and you're in the bathroom at the same time they are, if they don't wash their hands, don't eat there. Okay? Most of the time when a restaurant has an E. coli breakout, it's usually related to someone who's not been washing their hands after using the bathroom. Right? Because E. coli are present in feces. Okay, E. coli bacteria are always present. That's how they can tell if a water supply has been contaminated by sewage. If the water supply contains E. coli bacteria, it was contaminated by sewage. All right. So these ones, though, have different types of bacteria in their intestines that help to break down and ammonify, okay, the nitrogen available, okay, in the soil and in the atmosphere. Okay. Some plants, like beans, okay, they have nodules, little white things on the ends of their roots, okay? And inside those nodules live bacteria that can actually take nitrogen out of the atmosphere and turn it into nitrates, okay, that the plant can use directly. That's why a lot of farmers will cycle their crops, okay, or rotate their crops. They'll grow something like canola one year, and then the next year they'll grow beans, right, on that same field, because the beans will naturally put a lot of the nitrogen used up by the canola back into the soil without them having to put in a whole bunch of fertilizer. All right, so it's kind of a natural way to replenish. Okay, we're not going to talk about the nitrifiers here. Okay, nitrogen cycle, kind of what we've been talking about here, with nitrogen coming in and going out, and obviously being absorbed by the plants. Okay, now, what do you suppose that picture is of? It's a scanning electron microscope picture. Looks like a waterfall. What do you suppose all the little yellow things are? They're tetanus bacteria on the sharp end of a pin. That's the sharp end of a pin, okay? Under an electron microscope. All right? I mean, we look at a pin and we go, man, that looks really sharp on the end, but to an electron microscope that can blow it up a lot more, that's what it looks like on the end. Okay? Still sharp enough to pierce our skin, obviously. Okay? But look how much bacteria is on there. How many of you have ever used a pin to take out a sliver? Did you heat it up first? Okay? Or put it in alcohol? Yeah. Okay? If you're ever going to use something inside or underneath your skin, like a pin to take out a sliver, think of this picture. All right? That's how much bacteria is living on something that you don't sterilize at any time. Okay? If we were to take an electron microscope picture of a keyboard, you would barf. Okay? It is disgusting. Door handles, I'm going to turn you all into germaphobes here, but okay. Anything that is touched, okay, anything that is touched by a lot of hands is going to be covered in bacteria. Okay? One of the most disgusting things ever was actually a payphone. All right? Yeah, I know we don't use payphones much anymore because we all have cell phones now, but okay, back in the day we used these things called payphones where you put coins in them. Okay? And the mouthpiece, I mean, everyone's got that really close to their mouth and they're talking into it, and you always have a little bit of spray, right? Yeah. Okay? Pretty disgusting. Yep. Well, a toilet seat gets cleaned really often, right? Okay? I mean, if you're in a good bathroom. Okay? If you're in a public bathroom, okay, most of the time they've got sheets in public bathrooms, and it's like every hour someone's been in there to clean the bathroom, right? Okay, if you go like the Centennial Center's got that. Lots of fast food joints have that. You know, this last time this was cleaned was then. It's signed off by somebody. Okay, so those things are getting cleaned often. They're also, I mean, as much as we think a bathroom is really gross, okay, you're not touching it as often with your hands. Like, you know, like you might touch part of the toilet seat with your hands, but most of the time you're just sitting on it. Well, believe it or not, those parts of your body are cleaner, okay, than your hands. Because you're always putting your hands in stuff. You're always touching stuff with your hands. Your hands are disgusting, okay? Really, they are, right? They're covered, yeah. And if you're like me and you chew your fingernails, like, I can't stop because it's just a bad habit, but, I mean, there's just so much bacteria on there all the time. Okay, shaking hands with people. We actually had a few years ago when that whole H1N1 thing was going out. We didn't shake hands for giving the sign of peace. Everyone just gave the pound. Okay, they're safer. It's the outside of your hand. Yeah, elbows. Yeah, forearm bash. Okay, yeah, something like that. Okay, but yeah, it was no one wanted to shake hands, right? Like my grandmother, she knew this. I mean, she died a few years ago, but I mean, she would always, the expression when I was little, as soon as we got home from church, go wash your church hands before you eat. Okay, because you know you just gave the sign of peace to everybody, and you shook all these people's hands, right? Now you think of that. We give the sign of peace, then we go and get communion. Okay, but what did they used to do? Did they used to put it in your hands? No. You used to stick your tongue out. The priest would put it right on your tongue. Okay, does the priest wash his hands? It's part of the ceremony. Come on, right? His odds up there. They put the water on. Okay, they actually also now usually have some hand sanitizer up there, but okay. So these are all things obviously that we have to be aware of. Okay, things that we got to be conscious of in a germ-infested world. All right. So the next time you use a pin to take out a sliver, either soak it in some alcohol, or what I usually do is I just light a match or take a lighter and just kind of over on. Don't heat it up till it's glowing. Okay, you don't need that. You don't need that. Just run the flame over it a few times. That'll kill the bacteria, and you're good to go. Okay. Now, this is what your average, listening please, what your average bacterium would look like. Okay. Now this is a bacilli bacteria, but I mean they're all kind of the same. Okay. You'll see. You don't see all that stuff that you saw in the eukaryotic cell. Okay? There's no nucleus. There's a nucleoid region where the DNA is kind of snarled together. Okay? There's a membrane. There's a cell wall. Okay? There's ribosomes. That'd be about the only recognizable organelle that would be in there. You got some flagellum. Okay? And you have a mesosome, which does a couple of little things. But essentially, it's very simple. All right? And that, again, that compartmentalization that we talked about before comes back. This isn't as efficient, which is why you never see specialization happening or any multicellular prokaryotic organisms. Okay. All right, and DNA usually in a bacterium is in a ring, usually in a circle. So when it actually condenses down out of the snarl, it's a ring as opposed to many chromosomes. All right. Now, here's the other thing. Prokaryotes, because they're so simple, can go dormant. And they can go dormant for hundreds, even thousands of years. And then when conditions are right, start growing again. Okay? And it's believed that an endospore, which is a dormant bacteria, was responsible for the curse of Tutankhamun. How many people know who Tutankhamun was? King Tut. Okay, King Tut. When his tomb was discovered, yeah, most of the guys that went into the tomb or who were there when the sarcophagus was opened, they inhaled the spores because it's very dusty, right? You open up the sarcophagus, there's differences in atmospheric pressure, and dust blew out. Okay, they inhaled this dust, which contained the endospores of the bacteria, and they died. All right, probably from the same bacteria that killed King Tut. All right, but of course that added to the legend because at that time nobody knew about this kind of stuff. Okay, and it was like, well, you disturbed his grave, and his curse got you, and you all died. Right? Well, I mean, hey. It makes for a great story, right? But at least now we know why it happened. All right. Now, we've already talked all about eukaryotes. Listen up here, guys. We only got a couple minutes left. Okay, we've already talked all about eukaryotes, so we know kind of the importance of the membrane-enclosed organelles, okay, and having all those specific conditions, so we'll just kind of breeze over this. Now, how eukaryotes evolved from prokaryotes. Okay, couple of theories, probably a combination of the two. The eukaryotic cell is probably the result of some of what we call endosymbiosis, which means the absorption of other organisms into the cell. You have your main DNA, and then you have mitochondrial DNA. Inside of all of your mitochondria is a different set of DNA. Not as much of it, but there's a different set of it, which leads people to believe that these were actually organisms absorbed by very old kind of prehistoric cells, and they just kind of stayed there, and they ended up kind of growing together and serving each other's purposes. Okay, same with the chloroplasts. Okay, and there's also this idea that the cell membrane may have just what we call invaginated, which is kind of folded in on itself to create other organelles. Probably a combination of both processes. Okay, would have led to the development of eukaryotic cells with organelles and things like that. All right, we're going to leave it at that. Okay, remember, guys, cell anatomy lab due on Thursday. Make sure you got it done. I know a few of you have sent it to me already. Okay, and sorry, we haven't done the other one yet. We'll do it tomorrow. All right.